Assalamu alaikum students. Today our topic is Watson and Crick's model, that is a double helical structure of DNA model. First we see its definition, then we would explain it with the help of theory and diagrams. So students, before going to our educational topic, please subscribe my channel Muntazir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. So come back to our topic. In 1953, James Watson and Francis Crick, both these scientists were two young researchers in the University of Cambridge. Both these scientists were working in this Cambridge University at that time when they carried out double helical structure of DNA model. So during that time, when they worked out on the structure of DNA molecule, they made the basis of this model on the X-ray diffraction pattern. So first we shall take an overlook of X-ray diffraction analysis. A British chemist, Rosalind Franklin, carried on an X-ray diffraction analysis of DNA. In this analysis, a crystalline DNA molecule is bombarded with a beam of X-ray. When individual rays strike their atoms to the DNA molecule, their path is bent or diffracted, and the diffraction pattern is recorded on the photographic film. So this is the X-ray diffraction analysis diagram of DNA. Rosalind Franklin prepared this X-ray diffraction pattern of DNA molecule in the laboratory of British biochemist Maurice Wilkins, who prepared DNA fibers. This diffraction pattern suggested that DNA molecule had a shape of a helix that had a twist form. It is the first point of X-ray diffraction pattern analysis of DNA molecule. Number 2 point is the helix has a diameter of 2 nanometer. Its third point is a complete helical turn every 3.4 nanometer. So these are the three main points of X-ray diffraction pattern. The first one is DNA molecule had a shape of a helix. The second is this helix has a diameter of 2 nanometer, and the third is a complete helical turn every 3.4 nanometer. Now we come back to the important features of the DNA molecule according to Watson and Crick model. Its first point is that DNA molecule is a simple double helix, which means that DNA molecule consists of two strands. Both of these two strands are twisted each other to form a coiled shape like a rope. Nitrogenous bases are present at the inner side of the both strands. These nitrogenous bases are adenine, guanine, cytosine and thymine. Nitrogenous base of the first strand, that is any one of the four bases, we can say here is adenine, present at its inner side to form a pair with the nitrogenous base of the other strand, which is a thymine here. Thymine is also present at the inner side of the second DNA strand. So nitrogenous bases of the both strands form a pair which is known as nitrogenous base pair. It is so called because this base pair contains nitrogen in it. Similarly, cytosine forms a base pair with the guanine. So in a base pair, adenine always pair with thymine and guanine always pairs with cytosine. So in a base pair, both of the bases are oriented towards each other. A base pair is always purines and pyrimidines. Purines are large bases such as adenine and guanine. Both are double ringed and large bases, whereas pyrimidines are single ringed and small bases such as thymine, cytosine and uracil. All the three bases contain only single ring, so these are small bases. So in base pairs, purines, which are large bases such as adenine and guanine, directing towards pyrimidines, which are single ringed and small bases. So these base pairs keeping the Diameter of the molecule, a constant of 2 nanometer. A constant is that quantity that does not vary, because constant means a special number that representing a special quantity assumed to have a special value in a special context. This thymine and adenine is a base pair. So in between both of these nitrogenous bases A and T, there is present hydrogen bond. These two lines show hydrogen bond that is present between adenine and thymine. Similarly, in between guanine and cytosine, there is also present hydrogen bond. These three lines shows three hydrogen bonds that are present between the guanine and cytosine. So due to the presence of these hydrogen bonds, as two hydrogen bonds are present between adenine and thymine and three hydrogen bonds are present between guanine and cytosine, double helix DNA molecule is stabilized as a duplex. This duplex of DNA molecule is composed of two antiparallel strands. One strand is running from 5 prime to 3rd prime and the other strand is running from 3rd prime to 5 prime. By the earlier discussion, we shall conclude that a duplex of DNA molecule is composed of two strands. Both these strands are antiparallel. As antiparallel means both of the DNA strands run in opposite direction. If one strand run from 5 prime to 3rd prime direction, then the opposite strand will run in 3rd prime to 5 prime direction. In this double helix, adenine forms two hydrogen bonds with the thymine and guanine forms three hydrogen bonds with the cytosine, which means that adenine is always attached and bonded with thymine by means of two hydrogen bonds, whereas guanine, that is a double ringed and large base like adenine, will attach and bond with the cytosine, that is a single ring base and is a small base like thymine. So in between guanine and cytosine, three hydrogen bonds will be present. Adenine will not form proper hydrogen bond with cytosine because the complementary base pairing between the adenine and cytosine is not possible. But if adenine will bind to cytosine in any case to form a complementary base pairing, then it will lead to the mutation and cause a change in the phenotype of the organism. Guanine will not also form hydrogen bond with the thymine because the previous statement is also the same for the base pairing between guanine and thymine. Their base pairing is also not possible, but in any one of the case, if it is present, then it will lead to the mutation and become origin of the change in those organisms that carry such mutation or such base pairing. So because of this base pairing, adenine and thymine will always occur in the same proportion and same measurement in any DNA molecule as guanine and cytosine. The base pairs are planar and stacked, which means that the base pairs are arranged in a simple and smooth manner. These base pairs make an assemblage by joining themselves together to form their complementary base pairs. 0.34 nanometer apart, which means that these base pairs are 0.34 nanometer independent singly from each other as a result of hydrophobic interactions that contribute to the overall stability and overall establishment to the molecule, because by the arrangement of the base pairs that are smooth and in assemblage in a DNA molecule, the form of double helical DNA molecule is determined, and this figure of the DNA molecule is a uniform for all the cells of all the organisms and gives it a consistency as a whole. It is the unique property of a DNA molecule to holding its base pairs and help in retaining the DNA molecule shape. In addition to nitrogenous bases, DNA strands also contain a phosphate group and a pentose sugar. Both these phosphate group and pentose sugar form sugar phosphate backbone. When a phosphate group attaches to the pentose sugar, then it forms a phosphodiester bond as a complete helical turn every 3.4 nanometer. So a DNA molecule has two grooves. One is major groove and the other is minor groove. So students, it is our topic today, Watson and Crick DNA model. Keep your attention to your education. It doesn't only benefit you but your society, your family and also your country. Thank you for watching this your video.